Starting with the name of Allah, who is the most beneficial and most merciful. My name is Sameena Akhtar M Phil, microbiology second semester. My registration number is MB3 2022-2014. My course instructor is Dr. Niaz Muhammad. Today my topic of presentation is about the human genome therapy or human gene therapy. This is the outline of my presentation topic: introduction, types of gene therapy, approach in gene therapy, example of ex vivo gene therapy, application of gene therapy, and success story. So first of all, I will introduce my topic: human gene therapy. Human gene therapy is a cutting-edge field of biomedical research and medicine that aims to treat or prevent genetic diseases by introducing therapeutic gene into the cell of individual. This is also called gene transfer therapy. Gene therapy is a technique which involves the replacement of defective gene with a healthy one to treat genetic diseases. The fundamental concept behind the gene therapy is to modify genetic material within a patient cell to restore or enhance their normal function. This involves the introduction of specific gene into target cells, which can be achieved by using various delivery methods such as viral vector, non-viral vector, or genome editing techniques. In 1970, the idea of gene therapy is proposed by two scientists, Stanford Rogar and Richard Mulligan, who suggest using genetic material to treat diseases. In 1972, Paul Berg successfully combined DNA from two different organisms, marking the first step toward the genetic engineering. Now we will discuss about the types of gene therapy. There are two types of gene therapy. One is the somatic gene therapy and second one is the germline gene therapy. Somatic gene therapy involves modifying genes in the somatic cells of an individual. Somatic cells are non-reproductive cells that make up the majority of our body, including cells in organs, muscles, and tissue. The goal of somatic cell gene therapy is to treat specific diseases or conditions by targeting the affected cell directly. Scope: the genetic modifications are limited to the individual being treated and are not passed on to the future generation because somatic cells are not involved in reproduction, so that will be only limited to the individual, not to the next generation. Germline gene therapy. Germline gene therapy, on the other hand, involves modifying the gene in the germ cells, which are the reproductive cells, sperm and egg, and gave rise to the next generation. The objective of germline therapy is to introduce the genetic changes that can be passed on to the offspring. Scope: the genetic modification made in the germ cells are heritable and can be transmitted to the future generation. They will be transferred to the future generation because they are involved in reproduction, and reproduction during meiosis. They will transfer to the next generation, or they can be transformed from parents to their offspring. Now, approach in gene therapy. Approach in gene therapy are two types: ex vivo gene therapy and in vivo gene therapy. In ex vivo gene therapy, cells are removed from a patient's body and genetically modified in a laboratory setting before being reintroduced back into the patient. While in vivo gene therapy, the genetic material is directly derived into the patient's body, targeting the specific tissues or cells that require gene modification. This is the diagrammatically representation of ex vivo and in vivo gene therapy. In vivo, there are different types of viral vectors are used. AAV is adenosine associated viral vectors. LVS, it is also a form of vector. With the help of this, modifying tissues or cell can be transferred or introduced into the patient's cell, and they introduced to the patient's body. Now, example of ex vivo gene therapy. One example of ex vivo gene therapy is the treatment of severe combined immunodeficiency, also known as bubbly boy disease. It is a genetic disorder that impairs the immune system, leaving affected individual highly susceptible to infection. In the past, this condition was often fatal in early childhood. Nowadays, the excessive gene therapy, ex vivo gene therapy is most frequently applied to the hematopoietic stem cell that is used to treat blood immunological diseases as well as genetic diseases that affect tissue and organ. Now, application of gene therapy. There are a lot of applications in the gene which involve the gene therapy. Genetic disorder. It is also helpful in treatment of the genetic disorder, treating monogenic disorder like cystic fibrosis, muscular dystrophy, and hemophilia. The treatment of these diseases is also possible possible with the help of gene therapy. Providing functional copies defective gene or correcting mutation. कभी कभार म्यूटेशन आ जाती है या जीन म्यूटेशन होती है, तो उसकी वजह से जो डिफेक्टिव जीन होते हैं, वो म्यूटेटेड जीन होते हैं, उसको रिमूव किया जाता है विद द हेल्प ऑफ जीन थेरेपी, एंड सम हेल्थी जीन आर इंसर्टेड into the genome of individual. Cancer. Targeting cancer cells to inhibit growth or induce cell death. That is also done with the help of gene therapy. Inherited disease. There are a lot of diseases which are inherited or associated with the germ-line cells. That can also be possible with the help of gene therapy. Preventing genetic diseases from passing to the next generation, modifying germ-line cells to eliminate hereditary disorder. Now, success story. There are a lot of successes which are done due to the gene therapy or with the help of gene therapy. Severe combined immunodeficiency. First successful gene therapy in human restored immune function in patients with a faulty gene. Labor congenital amaurosis. This is also done with the help of gene therapy. Improved vision in children with a rare genetic eye disorder. Hemophilia B. Increased blood clotting factor level in patient, reducing bleeding episode. Challenges and ethical consideration. In the field of gene therapy, there are a lot of challenges and also some ethical consideration. Some of them are discussed below. Safety concern. Potential for adverse immune reaction or unintended genetic modification. Rigorous testing and monitoring are essential. High cost. Gene therapy treatment can be expensive, limiting accessibility. Continued research and technological advancement can help reduce costs. Ethical question. Controversies surrounding germline gene editing and hereditary modification, due to which in UK and some other countries this is illegal. Ethical guidelines and public diseases are necessary. Future prospects. Expanding treatment option, developing gene therapy for a wider range of genetic diseases, customizing therapy based on individual genetic profile. CRISPR-Cas9 technology is also a gene editing technique used in gene therapy, which are found in the archaea and also in the bacteria that act as in adaptive immune system, which are found in bacteria in archaea. Gene therapy beyond genetics, exploring gene therapies for non-genetic conditions like cardiovascular disease and neurodegenerative disorder that can also be that will be also done with the help of gene therapy. Conclusion. From all of these aspects, we have concluded that gene therapy holds immense potential for revolutionizing medicine and transforming the lives of patients with genetic diseases. While challenges exist, continued research in technological advancement and ethical consideration will shape the future of gene therapy. Thank you for your attention.